Hello everyone, let's begin the second lecture of Module 7. Here, I will explain what feminized seeds are. Feminized seeds are a seed bred to produce female plants almost exclusively, eliminating male plants. Cannabis is similar to humans, that each plant has a specific gender, male or female, that is designated genetically by an X or Y chromosome. So, male plant carries an XY genotype, and the female plant is characterized by the double X sex chromosome composition. S1Cs derived from selfing are also double X. So why do cannabis growers prefer feminized seeds? This is because feminized seeds have many advantages. So let's look at them one by one. Regular seeds produce male and female plants. Male plants release pollen that can fertilize females. Fertilized female flowers produce seeds, and the cannabinoid content decreases. Growers aim for sessile, but even one male plant pollinates the crop, reducing quality and yield. Expected yield drops by 30 to 50% due to male plants. This slide shows the principle of a feminized seed. Feminized seed will grow into female plants only. A genetically female plant that contains two X chromosomes is induced to form male flowers with pollen. Pollination with another female plant produce all female offsprings, double X. Pollen crossed with egg produce a double X seed. Feminized seeds are produced by inducing a female plant to produce pollen, which is then used to pollinate another female plant. This is achieved by spraying the female plant with a solution containing an ethylene inhibitor like colloidal silver or silver thiosulfate, which causes it to develop male pollen sacs. The resulting seeds have only female chromosomes and will produce all female offspring. This slide shows the techniques of feminized seed production. The mechanism is that silver ions bind to the ethylene receptors, such as ETR1 or ERS1, replacing the copper ion that normally facilitate ethylene binding. This blocks ethylene perception, which prevents activation of ethylene signaling genes. Then male flower promoting genes expressed. Subsequently, female plants produce male flowers containing X chromosome pollen. When female plants are treated with masculinizing chemicals such as silver nitrate and silver thiosulfate or phytohormone gibberellins, they inhibit production of ethylene that promotes female flowering. This suppression of ethylene production induces male flower production. Gibberellin induces male flowers in female plants. This ability can be used to produce feminized seed. For the feminized seed production, spraying GA3 solution for 2 to 3 weeks must be needed. After appearance of a male flower buds, within 2 weeks, they will be ripe to produce pollen in 2 another weeks. This slide shows the common mutations in cannabis, such as leaf morphology, chlorophyll production, growth habit, and sex expression. Let's look at one by one. Single leaf mutations show that leaves develop as a single blade instead of a compound leaf. Leaf splitting is a very common mutation found in skunk and equatorial varieties. Trilateral branching is another common mutation found in South American sativa strains. 3 branches are 120 degrees apart from one another. Twin seedlings mutation is that some seeds germinate to produce 2 seedlings from the same seed. Warping mutations are found in inbred lines and clones, usually seen in the leaves that curl sideways. Albinism is a lack of chlorophyll resulting in weaker growth and lower yields. Lastly, duck's foot mutation is that leaves are fused, resembling a duck's foot. It can offer advantages like camouflage. Polyploidy is a common phenomenon in plants, but polyploidy in cannabis is usually artificially induced. Polyploid cannabis plants may have more than 2 chromosome sets. Tetraploid cannabis plants have 4 times more chromosomes than normal diploid plants. For example, triploid plants have 30 and the tetraploid plant 40 chromosomes in each cells. This can affect various aspects of plant structure and anatomy, including cell size, leaf structure, and the root system. It can also affect growth and flowering rates. Polyploidy refers to having more than 2 sets of chromosomes created by disrupting spindle formation during mitosis. First, let's look at the advantages. Polyploid plants have increased stress tolerance. Tetraploid plants can be more resistant to adverse environmental conditions such as drought, poor soil quality, or pathogens. Polyploid plants contain higher contents of cannabinoids. Tetraploid plants can adapt better to different conditions and environmental changes, which can be advantageous for cultivation in different areas. Polyploid plants tend to be larger than diploid plants, which can be more challenging to grow and require more space. Some polyploid plants may provide lower yields, which can be disadvantageous to the grower. Problems in breeding. Difficulties may arise when trying to cross tetraploid varieties with diploid varieties, which may limit the ability to breed and create new varieties. Generally, as polyploidy increases, the amount of biomass also increases, and the amount of cannabinoids increases too. In cannabis, as polyploidy increases, various morphological characteristics appear. For example, larger leaves and thicker stems. The leaves show the larger stomata, enlarged cells, and the lower stomatal density. Additionally, the plants grow slowly and the flowering is also delayed. All right, everyone, let's stop here. Thank you. Bye.